Hi, I am Burcu Kotlu. Today I want to talk about our tool GLANET, which stands for Genomic Loci Annotation and Enrichment Tool. GLANET is a desktop application which is developed in Java. It comes with its graphical user interface. It can also run on command line. Its source code is available at GitHub and tutorials are provided at Docs. This is a joint work. Other than me, there are Can Fırtına, Sündüz Keleş and Öznur Taştan. Here is the outline of my presentation. I will give a brief introduction, continue with GLANET Overview, library representation, and I will go on with Annotation, Enrichment Analysis, Regulatory Sequence Analysis, and I will finish my presentation with case studies. Next generation sequencing technologies provides enormous number of genomic intervals. They represent genetic variations such as SNPs, insertions, deletions, or they can represent intervals of regulatory elements such as transcription factor binding sites, histone modification regions, and DNase hypersensitive sites. In order to interpret these intervals, we have to overlap them with known genomic and epigenomic annotations. Here we present GLANET as a comprehensive annotation and enrichment tool. This is the overview of GLANET. GLANET takes intervals of varying length and we accept different formats. You can provide the SNP IDs, you can upload your files in BET or GFF3 formats, or you can provide 0 base and 1 base coordinates of intervals. We first pre-process these intervals to eliminate any dependency within them. By using the intervals in our annotation library, we output the overlapping intervals as annotation output. When the given input consists of SNPs only, GLANET offers regulatory sequence analysis. We find the overlapping TF and SNP pairs, and we assess the effect of SNP on that TF binding. For enrichment analysis, we adjust for genomic biases such as GC content and mappability, and we list the enriched genomic elements and gene sets with respect to the given intervals. And here we show how we define gene-centric regions. We have defined exon introns and we have defined proximal regions as P1 and P2 in 5' and in 3'. We have also defined distal regions on 5' and 3' as 5D and 3D. Users can also extend annotation library by providing their own genomic intervals for genomic elements and for gene sets. And also we have another feature for examining gene sets. We examine them in 3 manners. Exon based, we only consider exons of genes. In regulatory based, we consider introns, 5' and 3' proximal regions of the genes. In all based, in addition to the regions defined for exon based and regulation based, we also consider the distal regions of the genes. This is how we represent our library. We store intervals in interval trees. Interval trees are in fact red and black trees, which are balanced binary trees. And for each genomic element type and isochore, we construct one interval tree. For example, for transcription factor element type, for all intervals of different transcription factors in different cell lines in chromosome 1, we construct only one interval tree. For annotation, we overlap the given intervals with the intervals in our annotation library, and we provide 2 options for quantifying this overlap. Existence of overlap just gives 1 or 0 depending on whether there is an overlap or not. Number of overlapping bases just gives the exact number of overlapping bases where each base is counted at most once. Enrichment analysis assesses the statistical significance of overlaps between given intervals and intervals of genomic elements in the annotation library. We use a resampling-based approach to obtain the empirical null distribution of the test statistic, and to evaluate the statistical significance of this test statistic, GLANET estimates empirical null distributions by randomly sampling intervals that match the characteristic of the given intervals. By default, we match the length and chromosome of the given intervals. Genomic biases, we adjust for genomic biases because the intervals that are derived from next generation sequencing experiments, because of these biases, they constrain the regions that can contribute to interval generation. And that's why we account for GC content and mappability with these 4 options, and we match GC content and mappability biases jointly or separately. And we do not require any input-specific file from the user. We can also match as a core family of the given interval. The idea is as follows. Human genome is divided into 5 regions with respect to their GC content composition, and they are named as L1, L2, H1, H2, and H3 with increasing GC levels. Joint enrichment analysis is valid for TF and KEGG pathways, and with this option, users can evaluate whether given intervals are enriched with a certain TF and with a certain KEGG pathway at the same time. Users can extend GLANET's default annotation library by providing their own intervals for gene sets and genomic elements. And this is how we carry out regulatory sequence analysis. In the first step, we find the SNP and TF pairs that overlap. In the second step, we generate sequences around this SNP locus. We take 20 base pairs upstream and downstream of the SNP locus, and in this way we generate reference and altered sequence or sequences. There can be more than one. And for control purposes, we generate a longer interval, approximately 400 base pair long, around this SNP locus. We also generate the position frequency matrices for this certain TF. Then, in the second step, we call the web service of an external tool called RSAT. We provide our sequences and the corresponding position frequency matrices, and RSAT returns us p-values in order to assess the effect of SNP on this TF binding. So, smaller the p-value, better the match is. Here's two results coming from regulatory sequence analysis of OCD GWAS SNPs. On the left-hand side, there is a SNP with this RSID, which has an overlap with STAT1 transcription factor. As the nucleotide changes from A to G, we have a better match with this STAT1 position frequency matrix, and therefore this is reflected with a smaller p-value for the SNP sequence. In contrast, on the right-hand side, for this SNP, which has an overlap with max transcription factor, as the nucleotide changes from C to T, it potentially disrupts the TF binding, and which is reflected with a higher p-value with the SNP sequence. And now let's go on with our case studies. In the first case study, we will make an analysis of OCD GWAS SNPs. First, let's load the input data. It contains one base coordinate, so input format has to be selected as follows. Let's make annotation with respect to our default library, KEGG pathways, and joint TF KEGG pathway analysis. Let's perform enrichment. I don't change the default parameters. Let's make 10,000 samplings, and let's give a name for this job. And let's run. And here is the command line arguments and information about this run. Let's continue with the second case study. This time we will see how the user-defined gene set feature is used. Again, we will use the same input file, OCD GWAS SNPs, but this time we will analyze OCD GWAS SNPs with respect to gene ontology terms, which is not loaded in our default library as gene sets. Let's continue. Let's stop this one. Let's rerun the GUI. Now, let's select this checkbox, user-defined gene set annotation. These are the demo data I have prepared for this feature. Let's load the files, gene symbol. Let's give a name. Description file is optional. I have prepared for this demo. Let's give a name. Let's run. Okay, this time we will make annotation and enrichment analysis of OCD GWAS SNPs with the gene sets in gene ontology terms. Let's continue with the third case study. This time we will see how we can use user-defined library feature of GLANET. We will use the same input data for sake of simplicity, but this time we will make an annotation analysis of this file with respect to transcription factor intervals defined in our demo file. Let's do that one. Let's stop this one. Okay. As I said, we will not use our default library, but this time we will extend our library using user-defined library feature. And here is the demo file I have prepared. Let's perform enrichment. Let's give a name for it. User-defined library. Run. Okay. And we can continue with our last case study. This time we will accomplish regulatory sequence analysis for OCD GWAS SNPs. Let's stop this one. Let's open the GUI. This time I will provide RSIDs which are stored in a text file. It can be, of course, 1 base or 0 base coordinates also, just to show it. So let's select the SNP IDs. For regulatory sequence analysis, we have to select transcription factor annotation. We don't have to do any enrichment, so don't spend time with enrichment. And we have to check for regulatory sequence analysis. And run. These job names are stored in output folder. When the GLANET finishes, there is a directory created with this name under GLANET folder output. And this ends my presentation. Thank you for your time. Thank you for listening. If you have any questions, please do not hesitate to ask me. You can reach me through my email. Thank you.